So how was break? Anybody remember break?
Yeah.
What? Anyways, so this week is finishing up the heart monitor. Has anybody gotten a heart monitor to work?
Yeah, a lot of noise.
A lot of noise. You got to filter aggressively.
No, nothing but
Did you
noise.
guys see anything yesterday?
No, nothing but noise.
We spent like 4.5 hours working yesterday setting up the whole Matlab program. We couldn't see anything through the eyes at all. On the oscilloscope, we just saw noise, and on the Matlab program, we just got straight lines. Like there wasn't even any components that were within the
So you have the gain set up to about 1,000 someplace, right, on the preamp.
maximum on the dip amp, and we also tried increasing the gain on the optocoupler, and we really could not. We couldn't see anything.
So
Is there a way to place the electrode on your arm?
well, the papers that are linked up say that you can put them along here with a separation of one active electrode on the inside, one active electrode on the outside of the arm. And
Electrode here, electrode.
but it seems like a better idea to put an active electrode here and then say a positive electrode here and a negative here, and the reference either spot doesn't matter.
We were doing bicep and like lower forearm.
Yeah, I would try and stay off of the big muscles because you get EMG interference. So if you're on the inside here, at least you're closer to the heart.
Down here, so we just have to.
Yeah, so you push in, you get bone there. So you're off the big bulk of the muscle, which helps with EMG rejection.
Oh, you were targeting the muscle.
I put it down.
The EMG signal like 60 hertz, and so if we're only looking at signals between like 1 and 20 hertz, then wouldn't we be blocking out most of the EMG anyway?
You certainly would be, and that helps quite a lot, yes. I would say that I think what Pan Tompkins says that you should set the filters to what, 5 and 25?
We started out with that, and I just dropped it down to 1 because we weren't seeing anything.
So I mean, 1 is okay. I doubt if you have much noise on the bottom side anyway. It's probably mostly high frequency noise. With a QRS complex of about 50 milliseconds, you would think that a lot of the energy should be around about 20 hertz. So having a cutoff of 25 is reasonable. Having a cutoff of 5 or 10 is going to, with a repeat time of 1 second, is going to cause significant differentiation of the signal. It's going to act as a differentiator. So you're going to start seeing artifacts that look like this. But that's okay, because the timing between this and this halfway point, half rise time, is still the heart rate. So even if it is highly differentiated, it's okay. So you set the filter up, you set the gain up. One possibility for testing is to put a test signal on it which is about the same size as you expect. So whereas before you had 100K, 100K, and what was it here, 1K? Drop that to 100 ohms or even 10 ohms. Measure differentially across here and make sure you can see something. The article, I have to go back and look at it because I don't remember what it said. The article that had 3 electrodes across here probably had a calibrated amplitude diagram on it. I didn't bring my iPad, so I can't look it up. I feel so lost in space. So there was one article, I think it was EMG from ARM or something like that. I can't remember the title even. It would be nice to know how big a signal they actually got. Was it 10 microvolts or 100 microvolts? But you should adjust this resistance, or perhaps the sum of these resistances, so that the waveform coming out of here is something like the size of the EKG you expect to see. And you may as well hook up the test circuit before you slap electrodes on yourself.
But what does it mean when you get like this big a noise and then you do see something like that, but then it's not regular?
What kind of noise is it? Is it 60 hertz noise?
We did figure that out. So you might imagine a profile like that, but then you overlap that with 10 times the amplitude of noise.
Right, so if it's white noise, if it's thermal noise, then you filter. Now, I don't believe that you're going to be dominated by thermal noise. I think that all of the random noise sources are relatively small. What you are going to be dominated by is either 60 or the 400 kilohertz from the power supply.
But shouldn't those both be dominated in 20 hertz filter?
Yes, they should. Yes, they should. However, I saw a very good counterexample today of the 400 kilohertz signal coming through. Did you figure that out, Akshay? What was going on with that?
Only 10 down to 15 millivolts, so I still can't get rid of it.
We were looking at like less than 10 millivolt peak to peak noise amplitude, and we weren't getting any signal.
That peak to peak, that's at the output.
Yeah.
So 10 millivolts, and at the input that would be then 10 microvolts because you had a gain of 1,000 or some such thing. And so the question is to look up in this paper how big a signal you expect to see, because I don't know whether you should have seen it or not.
Do you recall which paper it was?
Let me see the titles. I think it was Study of Single Arm Electrode Using Flexible Print Circuit. Almost English. Yeah, that's the one. See if you can get an amplitude off of the what they're talking about there.
It looks like their noise signal is like 40 millivolts peak to peak, and the extracted ECG signal is approximately comparable.
Yeah, but what's the input amplitude? What's the gain of the circuit? That's what I'm asking. So they're getting like a 40 millivolt signal out, but what's the gain? I'm guessing, given that the potential on the surface from here to here is something like 10 millivolts, that the attenuation by the time you get out here is going to be on the order of a factor of 10 or 100. So instead of 10 millivolts, you'd get between 100 microvolts and a millivolt. Did you find a gain in there any place? Okay. So that's my guess, is that 100 microvolts. But and putting the electrodes further apart should make the difference greater. I'm surprised that 60 hertz doesn't dominate everything.
There's no gain listed in this paper.
Is there a circuit?
No, it's really short.
Yeah, it's sort of in English. Yeah. So guess you need to look at some of those references and figure out how big the input signal is, and design a test circuit to test it.
I found another source that applies that it's 3 millivolts. That's a lot.
Well, that is a lot because that's only a factor of 3 attenuated from from the surface.
Actually, let me be clear. It says the QRS region, as an example, is approximately 30 plus minus 30 millivolts times 10 external gain. So the QRS region is plus minus 30 volts, 10 millivolts.
Plus minus 30 millivolts. Right, okay, so plus minus 3 millivolts, you said.
Right.
So that's bigger than I thought it would be. That's only a factor of 3 attenuation. That should be, well, that's still a factor of probably 30 smaller than you were measuring for the EMG. EMG is around something like 100 millivolts.
There's also another interesting sentence that says moving the sensors 30 centimeters further down the arm reduces the signal by about half.
So the half distance, so the E-folding distance down your arm is something like 3 centimeters. That says getting as close to the shoulder as possible is like a really good idea. At least one electrode, right? Up in here someplace or in the axilla. Try and stay off the deltoid and tricep if you can. I don't have any triceps, that's easy.
The arm, but
Actually, yeah, right in here someplace is probably a good bet. You know, it's where you push in here and you kind of feel more bone than muscle.
is it better on skinny people or chubby? I mean, like between the teammates. So.
What are you saying?
Less muscular people. You don't want to get noisy muscle.
So if I had to completely make up a story, what's the impedance of tissue? What's the impedance of subcutaneous fat versus muscle? I have no idea, although I expect that fat is somewhat higher resistance. Now, whether or not that matters in this case is a little unclear because you've got some bizarre boundary conditions here. You have some potential generator and some bizarre boundary conditions, and you're not loading it. You're not requiring very much current. I haven't done the circuit analysis, and I don't know what the answer is, but I doubt if it matters too much.
What about this backside here? Is that a good thing? Or are we?
Well, there's big muscle here. Most people have pretty good bicep. A lot of people have pretty good tricep muscle back here. On the side, there's relatively little muscle on the two sides. There's relatively little muscle.
That's the idea. Okay.
Try and get in there. Now, if you're just laying your arm on the table and you're not twitching, then you shouldn't twitching your arm. Yeah. Then presumably you're not going to get very much EMG contamination.
Oh, we were trying to do the opposite.
I mean, as long as you're not moving, it shouldn't matter too much.
Why did we get that noise? Because that was very strange. It's like very sharp peaks right next to each other.
Like that?
Compress them till they are next to each other.
Probably.
Kind of like
And what is the frequency? What is the frequency? Go for it.
So what we got for, let's see, this was the signal. We got, I mean, that went up to there like this.
So you have to look at the waveform and know the frequency of these. What was the frequency of these things? Was it coherent? Was it repetitive?
it was not. They were not uniformly spaced, at least for us. And then there would be like it would be like mostly uniform in amplitude, and then not uniformly spaced. Also, there would be like big spikes.
Yeah,
And
exactly.
sometimes it would kind of look like it was a signal. Like we had the same kind of thing like that, where the waveform would go up like this, a teensy bit, and there'd be like a big spike in the beginning of it, and it would look like it was regular for a second, but then it would just go back.
Yeah.
Yeah.
I mean, you would get like 3 pulses that you would see, oh, that looks like a heartbeat, and then would go away.
Yeah. We had the same.
So I have to see it. If the spikes are uniform and dense, it's probably the isolator power supply. Now you could swap over to a 9-volt battery. Swap over to two 9-volt batteries and disconnect the isolator power supply. And that would get rid of any of the oscillator artifacts. I've got lots of 9-volt battery connectors. I don't have any 9-volt batteries, but the campus store does.
We also had the signal going into Matlab, and when we looked at it on Matlab, it was totally flat. Except for, like, every once in a while there'd be a little glitch.
So what was
with our band test filter.
But what was the total voltage at the input to the DAC? What was your amplitude excursion?
5 millivolts.
Yeah, see, that's less than that's only one LSB, so you won't see anything. You need a signal coming into the computer of probably half a volt or higher. So you got to put, if it's not another amplifier, right, or a filter with some gain to get at least half a volt. The excursion, I think the default excursion on those is plus minus 5 volts. They're 12-bit A to D converters, so it's mapping 10 volts into 12 bits. 12 bits is 4096. So then each bit is worth 1/4000th of 10 volts, which is 1/400th of 1 volt, which is 2.5 millivolts. So to get a pathetic 10% quantization accuracy, you have to have at least 25 or 30 millivolts. And to get anywhere near, say, 5% accuracy or 1% accuracy, you're going to have at least a volt input. So I'm guessing you were down in the quantization noise. This might not work. That's okay. You'll learn a lot trying. Get the noise down. Yes?
With regards to this potentially not working, I know my partner Joe isn't here, but I know he's an exam on Thursday.
Yes, you're saying how much time should you reasonably put in on it?
Exactly. Because, I mean, I personally could spend the whole afternoon, like the whole night.
No, that's a perfectly...
I know that he can't.
It's a perfectly reasonable question, given that there's no endpoint on this that's quantifiable. And I would say this is a 2-week lab. The only assignment is to get this to work with a 2-week. It's a 1-week lab because presumably the circuitry should have been done for last week. That's right. It's a 1-week lab, and so 9 hours of work.
So
9 hours in lab.
If we spend 9 hours before Thursday and don't have it working, then it's okay for us to come into lab and not be able to demo it if we put in time.
Well, yes, although it would be nice to know what the boundary conditions are. For instance, can you measure not using your arm, but using a test circuit, can you measure a signal which is the same size that you expect to see? So put in a test signal. I mean, this gets rid of the biology. It makes only electronics. Put in a test signal. Show that you can see it in Matlab. I mean, if you could do that, and you can't record off your arm in 1 week, all right, we'll call it, we'll call it a failed experiment.
Is there a way we can generate a signal from Matlab, put it out, have an output through that go to the circuit and input it into?
Yes.
Is there a way we can create like a QRS wave?
Yeah. Would you like me to write out a program that generates a QRS wave? Oh, in fact, I gave you one in the lab. It does exactly this. It's a pretty cheesy QRS wave. There's no adornment to it or anything, but it has the right. The lowest? You mean how many bits?
Yeah. No, lowest voltage.
Well, that's equivalent to saying how many bits is. I think it's a 0 to 5 volt. But what you would do is put out full scale and then attenuate it using exactly this setup. Right? You'd put in a nice big waveform up here, and then attenuate it down to whatever the appropriate size is. And you can output 250 samples a second out of that DAC, so that's enough to do this waveform just fine. But I think for testing, the most obvious thing would be to set up a square wave coming out of the signal generator with a width of 50 milliseconds and a repeat time of 1 second. That's pretty close to the waveform you expect, plus or minus a couple of Fourier components. You could even low pass filter this a little bit, but just for hacking around, I would use and adjust this to 1 volt right here, and then you can easily calculate the attenuation. I'll try and go through those papers right after class and come up with a guess about what the amplitude should be. I should have that on the top of my head, and I don't. So you'll get an email right after class. Yes, at 1:45 I get to interview somebody by Skype. That's an abstract experience, where you can almost like see their expression. You know.
I'm making a lecture on samples. There's a client for this tutoring job in China, and so he gave like a lesson on derivatives through Skype.
Uh-huh.
For people in China.
That's very abstract. I did a class in New York City a couple of weeks ago by Skype. So I'm on the big projector, in front of the class, doing the Big Brother act, and I see the class back on my monitor, but I'm seeing the whole class, so I can't actually see their lips move. So I have no idea who's actually talking, unless they hold up their hand, which I asked them to do so I could get some continuity.
If we have a battery-powered oscilloscope, then we could actually hook it up with it to see what's coming in at different stages when we...
Yes, that's true. If you have an isolated oscilloscope, you could hook it up anywhere.
Do these oscilloscopes, can they run off of battery?
These oscilloscopes do not run off batteries. There is a variant of this oscilloscope that has a battery pack, but we did not buy that variant.
Do we have a single one of those? Because it would be...
I do not have a single one. I do have a microcontroller that simulates an oscilloscope in real time and runs and outputs to a portable television set. And I wired myself up with ECG, duct tape and copper wire. It was horribly crude. And then walked down the hall and watched my ECG on this. I do have one of those. It's pretty crude in terms of capabilities relative to a real oscilloscope. Bandwidth is 2 kilohertz, for instance.
Good enough for us.
It is good enough. So I will try and be in the lab tomorrow afternoon. I will be in the lab tomorrow afternoon. I'll be in the lab Thursday and Friday, of course. And let's see what these circuits look like. But this is a really quick thing to set up. I will send you the amplitude you'd expect. And let's see what happens. So is everybody set now to start on final project next week? Gabrielle, you had some questions, or you had some comments or something about...
I wanted to check my idea with you, make sure it was okay.
And we did. So, and the idea is to do a real-time mapping of visual field by shining a laser out into the environment and seeing when you can see it and when you can't. And the only safety consideration there is that the laser has to be eye safe for everybody else in the room. Interestingly, I Googled up LED safety or infrared LED safety and came up with a whole bunch of stuff from laser manufacturers about what constitutes a safe infrared laser. And this is difficult for your eyes to deal with because you can't see infrared, and so you have no automatic protection. If somebody shines a green laser in your eyes, you look away very quickly because it doesn't feel very good. But you can't sense infrared, so you don't look away. It just sits there and cooks your retina. You don't have any particular pain sensors on your retina, and so you don't know you're being cooked. And you'll end up with holes in your retina that never get repaired. So it's a really bad idea to look into an infrared laser, the bottom line. And there's several classes of laser. Class 3 is limited to what, 2 milliwatts, I think? Is that right? And at least, so one of the groups, PDF I read, had taken some 2 milliwatt lasers and measured the actual output, and the highest one was 60 milliwatts. So that's 2 orders of magnitude higher than it should be, and is in a clearly dangerous range. But you're going to be using visible lasers, probably red. So you can go to very low power, and they're still clearly visible in an environment like this. There's a guy over in neurobiology that studies vision who is red-green colorblind to the point where he cannot see a red laser on a PowerPoint. It just looks white to him. That's really colorblind.
So one potential issue I was thinking about this is that it's going to be kind of subjective because you're going to have to press a button or something when it goes out,
Yeah.
but you can't see it anymore. And I wonder how much your vision processing is going to affect that. Like if it's going to, if we're trying to measure the blind spot, is it going to fill stuff in a weird way or?
So you're going to shine the laser back and forth across the azimuth, probably not so much up and down, but back and forth.
We're going to, if we can, both.
Both, okay.
I was thinking you could get a fraction of a sphere for each eye that you could then use in Matlab superimpose, and then color map it with the maximum frequency that you can see. So you get
That'd be cool. I saw that done once with ping pong balls. Rather than make a big sphere, they cut a ping pong ball in half and popped one into each eye, and then shone lights in from different directions. It's called a Gansfeld. G-A-N-F-E-L-D. Gansfeld just means entire field, right? That's it. So the drill here is that you're going to ask the person to fixate on a certain point, let's say a laser directly in front of them or an X on the board or something of the sort, and then bring in another laser from the edges and ask when they can see it, and also what you can see or not see in a region like right in here, which is where your blind spots are, one on each side. And a couple of aspects of this. One is the blind spots for your two eyes don't overlap. So you get the blind spot for this eye, you have to cover this eye, and vice versa. So you have to have some way of automatically doing that. Second is that people are much better at detecting when something enters their field rather than leaves their field. So it's probably better to start a laser out here and bring it in until they can see it. Another aspect is that you will notice a blinking light at your periphery long before you notice a steady light at your periphery. So if a light is blinking slowly, you'll see it way before you see a steady color. And you can imagine the evolutionary use of this. Movement. If the tiger is galloping at you, it's probably a good idea to turn and look at it. Right. So flickering in the field, in the edge of your field, is probably means something fast is happening out there. You better look at it. And you may have noticed back in the day when you still had CRT monitors. Remember back then? If you looked, sometimes if you looked away from it, it tended to flicker more than when you looked directly at it. Remember that? Well, that's because you have faster flicker fusion at the periphery than you do in the center of your field. And so I think that's one thing you want to measure also is flicker fusion, which varies quite a lot from person to person, it turns out.
Is that because you have more rods in that direction? You have more cones?
Yes, that's partly it. In the center of your field, you have mostly cones, which are fairly slow response. And near the edges of your field, it's mostly rods, which are, by the way, not color vision. And on the other hand, near the periphery of your vision, the receptors are much further apart. And so you'd think that the sensitivity should go down because you don't have as many receptors per unit area. There's some sort of trade-off there. But you're still faster at the periphery. Any other aspects of final projects you want to talk about?
Do you have any pressure transducers going on?
Pressure transducers? Yes, I do. What range?
So I wanted to create a digital stethoscope, right? So I was going to get one active stethoscope from my parents. I wasn't sure how to change that signal into electrical signal.
So if you have a stethoscope head, then what's coming out, the head gets flexed by your body cavity. And what comes out of it is a pressure wave, as you said. The classic way of detecting the pressure wave is with a microphone. You have to have a microphone that's good down to DC or close to it to get the waveform, because it's a rather slow waveform. You can also there are also fairly low bandwidth atmospheric pressure monitors that are made for cell phones that are sensitive enough that they can tell when you've gone up a flight of stairs. They can measure the differential atmospheric pressure over one flight of stairs. That way they can map you inside the Mall of America, figure out what floor you're on. Right? This is important, I guess. And it doesn't work over the long term because pressure outside is varying all the time. But over the short term, as you walk up the stairs or take the elevator up, you can get the differential and deal with it. Is that sensitive enough? I don't have a good feel for that. It would have to do some calculation and figure out what the pressure variations inside this device are. The acoustic pickup definitely works, but it probably doesn't get the fundamental. It probably doesn't get the stroke fundamental. Rather, it's getting some other kind of noise like valve noise, which is higher frequency.
Does the heart rate increase when we take an alcohol to simulate the
Heart rate typically does increase when you drink alcohol, and in fact, in a person that drinks a fair amount, it increases just before you drink. Because your brain has learned that there's an anesthetic, there's a depressive effect, and is compensating for that. So your brain jacks your blood pressure and increases your heart rate so that when you take a big slug of alcohol and everything slows down, you're still functional. This is a learned response.
Is there some way we could link this? Somebody is
Is there a way to relate this to alcohol level? I don't know. That's you could look at the literature and find out. I'm guessing that the relationship is fairly loose. It may be good for a given person, but I'm guessing it's not very good across population. It's because people respond to so many different things with variable heart rate. You know, you can be running, you can be sitting in a movie and be frightened, or see a scene which is engaging, and it'll change your heart rate, your breathing rate. So I don't know how good you could do with that. It's certainly not as good as pulling the alcohol content off your breath, which is quantitative for blood alcohol.
How does that work? How does your breath relate to your blood levels?
Well, I mean, in some sense, the air that goes into your lungs is in equilibrium with the gases in your blood, right? That's how you get diffusion and you get oxygen exchange. To the degree to which alcohol is volatile, you get a partitioning across the lung, which is proportional to the concentration in the blood. Some proportionality factor. You also produce quite a few other chemicals. Most people produce a detectable amount of acetone in their metabolism, which they exhale. And most people produce a fair amount of hydrogen sulfide in their breath. You know, parts per million, or a few parts per billion. It's not a huge amount, but it's there. If it was parts per thousand, nobody would come near you. You'd smell like a rotten egg. But you certainly do produce a variety of volatiles in metabolism that are in equilibrium with your breath. For a short time after you take a drink, there's contamination by alcohol evaporating from your throat and esophagus. But once that's dissipated, the alcohol you exhale is very closely proportional to the alcohol in your blood. A breathalyzer is a good project. I've had people do that in 4760 as a microcontroller project. The challenge there is to figure out how they can ethically test it, particularly if they're under 21. There are chemical sensors which use adsorbed alcohol to change the potential of an electrode. So you can actually buy those sensors.
How long do those sensors last?
How long do the sensors last? They last quite a while. But I don't know whether that's 6 months or 2 years. I believe they're a polymer, and they've got some sort of ion exchanger on it, and there may be a chemical reaction as well. Alcohol dehydrogenase, who knows? But again, that you can look up. I could talk about chemical sensors if you want. Do a lecture on that. Anything else on final projects? Everybody's sort of settled in on a project. I put up a couple of versions of least mean squares filter code, one of which uses 2 taps to filter out 60 hertz, and a more general one that is a multi-tap. If you run it at a fairly high frequency, if you run the sampling at a fairly high frequency, let's say 5 kilohertz, and you ask for 50 or 100 weights, which become finite impulse response taps, then you can filter fairly broadband signals out, repetitive broadband signals. The two-tap filter, this is really easy to program, two-tap filter will cancel other things besides exactly 60 hertz. It does a pretty good job as long as, remember, I think I talked about this last time, you took the reference signal and you put it in a delay line and saved a sample from 4 samples ago, and that became the ref minus 90 degrees at 60 hertz. You need the reference in the 90-degree phase-shifted reference or something close to it so you can take an arbitrarily phase-shifted 60 hertz. After all, it's going to be phase-shifted if it goes through a low-pass filter. You have to take a relatively phase-shifted version of the 60 hertz and filter it. As long as you have a sample which is close to in phase and close to 90 degrees, you can do a vector cancellation. If you get less than 45 degrees between these two, you can't do the complete vector cancellation anymore. So the filter, which is designed for 60, works for 60 and a half and 59 and a half, and it even works for 62 or 63, but it doesn't work for 90 hertz. But the power line, I looked up the power line statistics, it's supposed to be within 1/10 of a hertz of 60 hertz at all times. So the two-tap filter should work just fine for canceling 60. But it doesn't sound like most people are having 60 hertz contamination. Although the LMS filter would work for any repetitive waveform, including the noise coming out of the power supply. Although it's too fast. Never mind, it's too fast. It's up in the hundreds of kilohertz range.
Would you take the power coming out of the power supply and would that be processed to the op-amps?
Oh, well, it's required. It's required that you have capacitors on the output, right? Yes, you could build a low-pass filter that looks like this, that goes off to the op-amps. However, for good regulation, for reasonable performance, this resistance has to be relatively small so that you don't have much voltage drop across here at whatever current you're drawing. So I would say the voltage drop across here shouldn't be more than a couple of tenths of a volt. And so you'd probably be limited to a few hundred ohms, which means this needs to be some monstrous capacitance. That's okay. You could try that. Big caps. But I've got some, like that. I could just see you dangling those off the board. That'd be amusing. Whole circuit's this big except for the capacitors, which you carry on your back. But what's more likely, if you're going to mess around like that, I would say for this lab, given the short time, use a pair of 9-volt batteries. Walk over to the campus store, get a pair of 9-volt batteries, hook them up, you got plus minus 9 volts, it's dead stable DC, end of story.
And even if there's a voltage drop, because it will be the same for both the terminals, so it will be okay.
In which? For this?
When we use a battery. So even if the voltage drops?
Even if the voltage drops from 9 volts? It won't, though, for quite a while. Your total current draw for the INA 121 plus 1 op-amp, plus the infrared emitter. The infrared emitter is sitting at around 10 to 30 mils someplace. 30 mils. So you figure that even a fairly cheap 9-volt battery should work for about 10 hours. They're good for about a half an amp hour. So 500 milliamp hours. So even if you're drawing 50 mils, they should be good for about 10 hours. That's good enough. What else?
The other day when you were talking about this lab, you said something about using Hilbert differentiation as part of the filtering. And I found a paper where they, I didn't know what that was, so I looked it up. And I found a paper, I couldn't find anything about Hilbert differentiation, but I found something about Hilbert transforms. And randomly, like the fourth hit down when I typed Hilbert differentiation into Google was ECG recognition totally by coincidence, where they took the derivative and then they also used the peaks of the Hilbert transform to constrain where you could find the peaks of the R complex.
So you want to know what
we're talking about?
So
as opposed to using just the diff function.
the diff function is a first order differentiator estimate, right? It takes the difference, so it's x n plus 1 minus x n. You could imagine that a little bit of smoothing would be a good idea. So you could take 3 samples before, average those together, take 3 samples after, average those together, and take the difference of those averages and get another estimate of the first derivative.
So the paper that did the differentiation and then the Hilbert transform also used the SPAPS function, which is a cubic spline function, to smooth out the waveform.
Right. So so the next step is then, rather than saying, all right, I'm just going to average these together, why don't I fit a curve through these, which is optimal in some sense, like a cubic spline, which is continuous through the second derivative. Continuous through the second derivative. And then calculate the derivative analytically from the shape of the cubic curve, which you can do in closed form. So you fit the cubic curve, and then you take the derivative at some point analytically, and you can get a different kind of average value. A Hilbert differentiator is just a waveform of a specific shape that you fit to the points before you that is then used to analytically calculate the derivative.
So if we do the cubic spline and then do the derivative, then that is a Hilbert differentiator.
That is a cubic spline differentiator, and it's quite good. It's probably as good as you need for this lab.
And so a Hilbert differentiator we do
Is just a different. No, it's just a different curve. It's not a Hilbert. Hilbert transform is yet another orthogonal transform. It's like a Fourier transform, or a Walsh transform, or there's a dozen different Laplace transform. It is yet another orthogonal transform, and I can't tell you what the basis functions are off the top of my head. I can go look them up.
Like 1 over pi something.
So it's some sort of fractional function.
Yeah.
So it's probably, yeah, all right.
What they use it for in the paper is they said the peak of the QRS complex would be would necessarily there would be one in between every 2 peaks of the Hilbert transform. And so if you know the timestamps of the Hilbert transform, the peaks in that, then you could use that to limit the range where you would find the QRS.
I can't talk about this right now, but I can look it up and talk about it. Generally speaking, the reason you use orthogonal transforms is that depending on the kind of waveform that you're transforming, you can concentrate energy into one or a few places in the transform space. Just as with a sine wave that goes on forever, you could put all of that energy into exactly one frequency in Fourier space. And a square wave, when transformed with a Walsh transform, puts all of the energy into one one component of the Walsh transform. Different orthogonal transforms compress energy differently. And you want to choose the one that gives you the optimum compression, the optimum signal to noise ratio with the minimum amount of work. Discrete cosine transform, which is used everywhere. It's used in MP3 encoders, JPEG encoders, is used because it tends to concentrate the maximum amount of energy in the lowest frequency components. That means that you can just throw away the high frequency energy and save lots of bandwidth. The main reason for that is that what's usually used is an 8-point or 16-point DCT. That's a very short time discrete cosine transform. The signals of interest, audio signals, over 8 points tend to go like this. They tend to be sloped up or sloped down. It's a small part of a waveform. The fundamental of a Fourier transform over this same region has exactly one cycle in it and therefore cannot represent a gradient. It has an average value of 0. The fundamental of the discrete cosine transform, fundamental, looks like that. It's a half a cycle. Therefore, it encodes a linear gradient with one coefficient. And therefore, you save a huge amount of encoding effort if you use a discrete cosine transform rather than a Fourier transform for slowly varying signals, like images and sound. Which transform happens to work depends on the signal. I know that Hilbert transforms are very good at compressing space-time into dots. They also produce some really weird artifacts relative to Fourier transforms, but that may not matter for recognition. What else for anything else on final projects? So I was thinking about going back and talking a little more about action potentials and hearts and the heart models, the transistor models, which are kind of simulate channels, as one possibility for a lecture or two. And I used these in a lab, freshman lab last year, which worked pretty well. Another possibility is to start talking a little bit about timing and oscillators as just a piece of instrumentation. How do you build an oscillator? What does an oscillator mean? How does biology do oscillators? The heart's an oscillator. It's a pretty crummy oscillator. It has a lot of variability, but it sits there and oscillates.
Can a machine be programmed to actually, following on what Manish is trying to make, is making a digital subscript? So I don't know what doctors look for when they hear.
Oh, can you make a device that does automatic recognition of a normal versus an abnormal heartbeat? Yeah. And the answer is maybe. Humans are awfully good at pattern recognition. On the other hand, neural networks and various other kinds of machine learning systems don't have the finite limitations of ears. And there are some neural network, there are some combination analyzers. You put some sort of analyzer on the front end, Fourier analysis, Hilbert transform, whatever it is, Mel-Sepstrom analysis, whatever. You put some sort of analyzer on the front end, and then you take the output of the analyzer, which has a bunch of time-varying coefficients. You put that through some sort of recognizer, which may use machine learning, or it may use patterns that human programs in. And then ask, is the output of that recognizer better or worse than a human who is trained to do the job? And I think, I have to go look this up, but I think the current state is that ECG recognizers are about as good as humans.
What about the lungs?
For lungs, couldn't tell you. Easy to look up. You'll probably be shocked at how many papers there are. I know that machine learning has been applied to ECG recognition, lung tumor recognition from MRI and CT scans. It's been used for trying to understand complicated sets of many tests all at once. So you do a CT scan, you do an MRI, you take blood chemistry, you do a urinalysis, you have the person run on the treadmill, you get a pile of data, and what you'd like is a diagnosis. And right now, I think the state of the art on that is the computer Watson is being trained to be an oncologist, I believe. If you can do Jeopardy. So they have a bunch of oncologists training Watson. Okay, Watson, what is this? Oh no, you're wrong. It's just, yeah, and they're training the machine until it becomes an online diagnostician that anybody will be able to use. And that will happen. Humans are very computationally good at recognizing things, but sooner or later, you're just overwhelmed by any machine. You know, see how John Henry and the steam hammer, right? You just, no way you're going to drive more spikes than a steam hammer. Why should you expect to recognize more action potentials than a computer? If you wanted to do something with machine learning, that'd be all right. There's support in Matlab for neural net fabrication. You have to figure out some front end for doing the analysis, because typically the time series is too high dimensional. What you need is some way to get the dimensionality down, which is what a Fourier analysis or some other spectral analysis does. And you may look for invariance in that spectrum. And the analysis system may look for invariance before it's been put into the recognizer. For instance, there's a scheme for recognizing humans by their voice. How do I know it's my mom on the phone? It's not by what she says, it's by how she sounds. What do I mean by that exactly? What is it that is invariant about her voice that's independent of what she says or how mad she is that allows me to detect who it is? A lot of work's been done on this because human recognition is important. And the scheme that seems to work best, and this is really abstract, you Fourier analyze, you do short-term Fourier analysis of the voice, and then you compensate the Fourier spectrum by human sensitivity, which is called a MEL compensation, MEL compensation. And then you take then you do a discrete cosine transform of the FFT. What does that mean? And then the coefficients of the discrete cosine transform of the MEL adjusted FFT has components which are related to identity. And then you can train a neural net to recognize, say, the lowest 12 coefficients of the discrete cosine transform. the DC coefficients and the first 12 or so harmonics. And it's pretty good. What does that mean? I don't know. I have no intuition for that at all. Somehow the DCT of the FFT is measuring something about periodicity of the FFT. Right? It's measuring something about where the peaks are in the fast Fourier transform. But I don't have any intuition for what it means, but it works. Yes?
I bet they did find out they did like a million transforms and it had like a whole bunch of features and then ran a classifier on random subsets of those features like a billion times and then found out which one worked.
You know, that might have happened. And on the other hand, it might have been some crazed graduate student who says, I'll bet that the periodicity of the peaks of the Fourier transform encodes some useful information. I'm going to try this. I got to graduate. You know, sound familiar? Yeah, yeah. So I don't know how that happened. It would be an interesting piece of history of science to try and trace that back to the first paper, which is not that long ago. So it's probably doable. And find out what their motivation, why did they try this crazy thing?
And why this crazy thing?
Yeah, say again?
Like what determines your voice sounds different than my voice?
So,
Just like the frequency of the waves coming out plus their amplitude?
so the simple model is that this is sort of the linear model, which is actually a pretty good model, is that you have some sort of waveform generator, your vocal cords. Everybody's vocal cords are a little different, so the power spectrum of this is slightly different in different people, maybe a little bit sharper peaks or a little bit broader peaks, maybe a little bit of growtiness on the falling edge because you've got a polyp or something on your vocal cords and they're kind of banging together a little bit. But you have some sort of driver function which is then put through a linear filter, which is your throat and nose. So you can model this system as a series of tubes of different diameter with a side tube coming off, which is your nose. And you can show that you can get decent sounding voice with about 10 resonant filters. So if you build a buzz generator and drive about 10 filters with it, you can get something that sounds like voice. And it's the shape of your throat, the length of your throat, the shape of your vocal cords, the amount of energy that comes out your nose rather than your mouth, that determines what you sound like. And you don't even have to have the driving element here to figure out who it is, because you can pretty much tell when somebody's whispering who they are. And that's now a white noise source. It's a hiss source from someplace down deep in your throat, which is then being shaped by this same filter setup, but with a different drive. And you can do various things like put different frequency drivers on your larynx. This is not awfully comfortable, but you can drive your larynx and get different sounds out of your mouth and ask, does it sound like you? Does it not sound like you? So it's some combination of this linear filtering system plus the driver. And very early, AT&T, maybe in the 50s, 40s, was doing the thing called a speech vocoder, which derived the fundamental of the voice, analyzed the 10 resonances of the throat, and merely transmitted those resonances and the pitch, and not the waveform. It transmitted, and these resonances change rather slowly with a time constant of about 10 milliseconds. So you can get a compression ratio, of phenomenal compression ratio, like 150 to 1. If you just change, you transmit the differences in the resonances every 10 milliseconds rather than the waveform itself. Does it sound like the original human? Nope. It sounds like a robot talking over the phone. Nobody likes it. But it sure is low bandwidth.
I think there's an experiment where, like, they take a recorder and record someone says, like, a bunch of days, right? So they have a huge amount of data, what this person say, a bunch of words this person says, and so on, and sort of analyze what every one of those have in common. Because, for instance, if you say anything, regardless of what word you say, I'm going to is your voice, right? So every single word you say should have something in common with each other, so that I can distinguish that, oh, it must be this.
That's right, and it has something to do with the Mel Sepstrom. The Sepstrom is spectrum spelled backwards or inside out or something. It's the Mel Sepstrom. It's this discrete cosine transform of the power spectrum. And whether you can get these from waveforms from time domain, I have no idea. I expect that the features are so spread out in time that they'd be very difficult to analyze. I don't know. That you can easily, there's been a pile of work done on speaker recognition.
In terms of truth, though, the only thing we receive is, because the sound wave goes through here, there are different terms that vibrate different resonant frequencies, right? So I think you as a human are detecting from the sound wave is the resonant frequencies, over here, right? So that's what's going back.
Well, that's not entirely clear, but approximately what you're doing is taking the Fourier spectrum of the sound coming in, but since Fourier transform is a linear transform, that maintains all of the information from the original signal if you maintain the phase. It turns out humans don't really maintain phase very well. So, so you are more or less doing a short-time Fourier analysis, and then reconstructing information from that. Now you're not. It's not exactly that. There's a really cool website in which they take the immensely complicated sounds that come out of a human and reduce it to 3 sine waves. So you take the energy, you take this massive energy, it's all over the place, and you do a chi by eye, or you do least squares fit, and you say, that's the 3 highest intensity peaks. I'm going to fit a sine wave to these 3 frequencies and then track those through time. So what you get is a, and then you get a second sine wave. You put 3 sine waves together and you can understand the speech. You put 5 together and it's very clear what's being said from just 5 pure tones that are tracking something about the dynamics of the system. It doesn't sound like anybody in particular anymore. It doesn't have enough spectral components to figure out who's talking, but you can understand the words. And, you know, nobody knows how it works. I mean, so if I don't hear anything about ideas for lectures, I'll start talking next time more about action potentials and cardiac models. Okay.
If any work's been done with like vestibular augmentation for balance.
Yeah, interesting. There has been some work done.